Hello, everyone. My name is Jason Kilgore. I'm with Molecular Probes Tech Support. We're part of Life Technologies. I'll be your presenter today. This deck was also made by Ed Lieber. He and I are both with Molecular Probes Tech Support. I've been with Molecular Probes now for 13 years, including 8.5 in R&D, making some of these products. Ed has been here for 12 years. And for those of you attending live, you can submit chat questions, and Ed will be answering those chat questions live. For those who are viewing the recording of it, there will be a slide at the end to contact Molecular Probes Tech Support. Also, I should preface by saying that there will be some survey questions that are pushed to you a little bit later on. We appreciate you taking just a moment to answer one question every now and then. Okay, let's go ahead and get started. So this presentation is designed to give you a very basic understanding of 4 of the main classes of fluorescent reagents, or fluorophores. I'll start by saying a little bit of a brief history on the beginning of fluorescent dyes and use with biological materials. And then I'll talk about the strengths and weaknesses and characteristics of each of those 4 classes. Organic dyes, Phycofluors like phycoerythrin, fluorescent proteins like GFP and RFP, and quantum dots, which a lot of people don't know about because they're fairly new. And then finally at the end, I'll say a few words about choosing your fluorescent reagents and matching them to your instruments. Some of the questions that this presentation is meant to answer, things like what are the limitations of each of these classes of dyes? What are their strengths? What are their weaknesses? What instruments are they useful for? For instance, why is RPE mainly used for flow cytometry and not microscopy? Under what circumstances would you use fluorescent proteins? And how are quantum dots different from all of these others? And then finally, how do you choose your dye and match it with your assay? First off, just a basic question, why dye your cells at all? On the left, you can see a typical transmitted light image of some cells. This one is contrast enhanced with a DIC method. And you can see you cannot really make out many structures using transmitted light. So instead, you want to label them. The old-fashioned way was to use colored dyes that don't fluoresce, like hematoxylin and eosin, especially for tissues. But a better method is to use fluorescence. As you can see in the image here, you get much better signal to background because you have a black background. And you're able, using filters, to select only the dye of interest, and that allows you better ability to multiplex, to add multiple different labels on the same sample and view them together. A few words about the discovery of fluorescence. The very first noted publication of a fluorescent dye was way back in 1830s. A preacher by the name of David Brewster in Scotland had taken, I think, laurel leaves and crushed them up in a solvent and extracted from them their pigments. And he noted that when the sunlight shone through the solution of pigments, he got a fluorescent red color. He didn't know what it meant, but that was the first instance, and that was in 1833 or 1834. It wasn't until 10 years later that this phenomenon was studied. Sir John Herschel, and then not long later George Stokes, looked at a solution of quinine sulfate, which was used as a malaria treatment. Quinine was extracted from the bark of a tree in Africa and was administered to people suffering from malaria to treat their disease. It's a very bitter solution, so they would add it to sparkling water to help it go down a bit. Later, gin. That's where our gin and tonic comes from. And he noticed when the sunlight shone through it, you got this, as he called it, a celestial blue fluorescent color coming through it. And this was the fluorescence of the quinine. And George Stokes really did the photophysical determination of this. And as you can see in the picture, if you shine a blue light into this solution of quinine, you get this beautiful blue color. You can buy tonic water with quinine sulfate in it at your local store, and do it yourself with just a keychain lamp. And what they noticed was that when the light shone through, the light came out the other side the same wavelength. In this instance, it's a laser, and green goes through the solution and comes out the other side, but the volume of solution that it passes through gives off a wavelength at a higher color, a higher nanometer wavelength. So green goes in and yellow comes out. What's going on there? And of course, in all of this discussion about fluorescence, we're talking about the visible wavelength mainly. That's what most assays utilize, anywhere from 400 nanometers up to 700 nanometers, and sometimes beyond that into the infrared. Those are the wavelengths that we're interested in for biological imaging. So when George Stokes looked at his quinine sulfate, he began to start to understand, for instance, that when you excite, a longer wavelength comes out the end. Later in 1834, Alexander Jablonski studied it as well, and he found that when you and he came up with this diagram that describes the fluorescent process. On the left-hand side, there where you see number 1, that's the excitation state. So when you shine a photon of light on a molecule of a fluorescent dye, you get this excitation. It's very fast, femtoseconds, and it raises up to that excitation level. Number 2 there, that's a relaxation state. So it relaxes, it loses a little bit of energy. That takes only about a picosecond. And then what's really important for our observations is number 3 there. That's the fluorescence. It loses energy in the form of a photon of light. And so you have energy going in of a higher level of energy, lower wavelength, and then a loss of energy in the form of a proton or photon at a higher wavelength, as number 3 there. And so this observation that a wavelength that goes in is emitted at a higher wavelength is the Stokes law, named after George Stokes there. And what you see here is the spectrum that's typical from fluorescent dyes. It's a bell curve for the most part, except for quantum dots, which I'll talk about later. So you have a curve on the left that represents the excitation wavelength of the dye, and then a curve on the right which represents the emission wavelength of the dye. So anywhere within that left bell curve, you can excite the dye maximally at that line where it says number 1, and anywhere on the right, you can catch the emission of the dye maximally at where it says number 3. This dye is Fluorescent 488, so it excites maximally at 488 nanometers, emits maximally at 520. And that difference between the peak excitation and the peak emission, that's the Stokes shift. The bigger the Stokes shift, the better, because this allows you better multiplexing and less overlap of the excitation emission between that and other dyes. Now let's talk a moment about the structure of dyes. On the left is phenolphthalein. This is a pH indicator that many of you are surely familiar with. It's non-fluorescent. It doesn't fluoresce. And it's a flexible structure. There's a lot of movement in this molecule, so a couple of those phenol rings there actually rotate around the bond. If you immobilize that bond and make fluorescein out of it, which is a very common traditional green fluorophore, an organic dye, it forms a rigid planar structure which allows better photophysics and an ability to emit at a fluorescent wavelength. So you'll see that when you look at the dye structures, they always have this planar structure and a lot of these phenol groups, phenol rings in it. And you can also see here the size. It's about 1 nanometer. Molecular weight of several hundred daltons up to maybe 1,200 daltons. So let's get into the first class of fluorophores, the organic dyes. These are really the first that were used, dating back to the late 1800s. Organic dyes are synthetically derived organic molecules that are used for all sorts of different fluorescent detection. Some of the earliest were fluorescine and rhodamine, acridine orange. Just a little bit of back history. I mentioned how quinine sulfate, everyone was trying to make quinine, a synthetic quinine, to treat malaria. One of those early people was a fellow by the last name of Perkins, who was trying to make a synthetic quinine. He wound up with this black goo. He tried to wash it out with a solvent, and in the solvent came out this beautiful reddish color. He dipped his white silk handkerchief into it and wound up dyeing his handkerchief this beautiful color. That color became mauve, and within a year he had formed the very first synthetic dye manufacturing factory in England and was dyeing Victorian ladies' clothing with this mauve color, and later developed other aniline dyes that were similar in structure that were blue and green and so forth. Another offspring of this attempt to make synthetic quinine were some of the first fluorescent dyes used to treat trypanosomes, for instance. And in the first fluorescent microscopes, they looked at these treated trypanosomes and saw fluorescence in the nuclei. That was acridine dyes, some of the predecessors to acridine, nucleic acid stain, which has an orange color. So it's an interesting tie-in with malaria there, with the history of dyes. And since then, they've taken these, companies have taken these dyes and added structures on them, functional groups and pendant groups that improve their characteristics, make them brighter, change their wavelength to blue or far red or red, make them less pH dependent, for instance. And then finally, companies started making specialized dyes with all of these, this knowledge of functional groups and their effects, such as the Fluorescent dyes, Oregon green dyes that we sell here at Molecular Probes. In this next slide, you'll see a range of some of these dye structures. At the top are 4 green dyes that overlap. They're all typical green, like fluorescine and wavelength. You can see that fluorescine, Oregon green, and Fluor 488 are very similar in structure. That's because our chemists have gone in and added those functional groups on them to make them more photostable and to give them improved characteristics like pH and sensitivity. But there are other dyes, like FL, for instance, which have a very different structure. On the bottom, you can see coumarin, which is a very typical blue dye, and Fluor 594 in the red. Notice that the more red-shifted the dye, the larger it tends to be. We can see that in this next slide as well. By the way, you'll be getting a survey question. Thank you very much for answering it. You'll see that Pacific blue, when you go from blue to green to red of 594, generally the dye structure increases in size and molecular weight. Also, the extinction coefficient tends to increase. The extinction coefficient is the measure of absorbance efficiency of a dye. The more it absorbs, the brighter the end fluorescence will be. Of course, a lot of companies now have some commercially available dyes that are optimized for various qualities. Here we feature our Fluor dyes. You can see that it's a full range from blue all the way up to infrared or near infrared. And our Fluor dyes, for instance, have very good photostability, meaning that they don't bleach as fast or go away as fast when you shine the light on them, which is a common problem with organic dyes. Our dyes are less pH sensitive and have higher excitation or quantum yields, which is a measure of the emission efficiency. And they're matched to a common laser line. What do you do with these dyes? I mean, some of the dye structures will, in themselves label live cells, for instance, go in and label nucleic acids or certain organelles like mitochondria. But many times you want to functionalize these dyes by adding reactive groups on the surface of the dyes so that they can react with certain functional groups on your molecule of interest. Make a conjugate. Conjugate is where you put the dye onto it covalently, onto your compound of interest. On the left here, for instance, you can see a phalloidin, which is a peptide, conjugated with one of our Alexafluor dyes, Alexafluor 88. This is one of my favorite reagents. The phallotoxins, like phalloidin, come from a deadly mushroom called the death cap mushroom. If you eat it, you die a horrible death because the peptide binds to the F-actin in your liver and wind up with liver failure as you get a transplant. However, with labeling cells, this is a good thing because the peptide binds to the F-actin in your cells, and you see the cytoskeletal structure quite nicely. Here in the middle, you can see a picture of a flatworm that's been labeled with this conjugate. You see the actin in it. On the right, you see another derivative of fluorescein in this case, where we've put on a 12-carbon tail, and that carbon tail then inserts into the membrane, this reagent, into a lipophilic probe for plasma membrane labeling. So there's a lot of things that you can do to derivatize these molecules and make them useful in specific assays. One common conjugate that you'll see is labeling of antibodies. There are a lot of primary antibodies that are labeled or secondary antibodies that are labeled. And I talked at length about this in some previous webinars for immunohistochemistry labeling. But you'll see here a representation on the left of a primary antibody that's labeled with dyes, and then that primary is used to detect a protein of interest or an antigen on your cell. You can see that if you use a secondary antibody to detect the first one, now you have more dyes present in a brighter signal. Of course, you have to be worried when doing antibodies about species cross-reactivity and nonspecific binding. But what I want to focus on here is more about the dyes. When you conjugate a dye to anything, you risk potentially inactivating the functional binding site of that protein. So that's one potential drawback of doing conjugation. Another one, if you have too little of the dye, you wind up with a conjugate that's too dim. If you add too much, you can sometimes have dye-dye quenching, where the dyes actually quench each other if they're too close and too crowded. Or you change the hydrophobicity and charge on your reagent, causing your protein to drop out of solution, precipitate, often irreversibly. So caution has to be taken when doing conjugations of this nature. I should add here that the next webinar in this series will be for our antibody conjugation kits. So be sure to check in for that. So there's a wide range of organic dyes used for all sorts of different assays. Here's an example image of where we've taken wheat germ agglutinin, which is a lectin, and labeled it with a red dye, Alexafluor 594. And that, in live cells, will bind to sialic acid residues on the cell surface, giving you this very nice plasma membrane stain. The green here is a GFP construct for mitochondria. The next image, another example. These are muntjac skin fibroblast cells. Muntjacs are these funny little deer that live in Indonesia. And their skin fibroblast cells have these amazing cables of actin that run across them. They're quite striking, I think. And here's that phalloidin that I talked about earlier from the death cap mushroom, right, labeling those actin cables. And in the green, we have an antibody, antipaxillin, which is a cell adhesion molecule, and an antibody against it that's been labeled with Alexafluor 488. There are a lot of nucleic acid stains also. DAPI is the most common one. You can see it here labeling the nuclei in blue. There's a lot of nucleic acid stains. Some are more RNA selective. Some pass through membranes to label live cells. Others you have to fix and permeabilize for it to get in, and those are often good dead cell indicators. We had a previous webinar on viability stains. I talked about use of those. So how do you choose an organic dye? I'll talk at length a little bit later about choosing dyes, but here's a sneak peek here. First off, what is the capability of the reagent? What structures do you want to label? Are you using an antibody? What dye is best for that? Think about your detection capabilities. What filters do you have on your instrument, and how do they match with the dyes? And I'll talk a little bit later about some of the tools you can use to match those dyes. The spectral viewer is one of those. There'll be a link here, but I'll show you what that looks like in a minute. So what filters do you have? What is your light source? If you're using a laser-based system, what is the laser wavelength? And does it match the excitation of your dyes? If you're multiplexing, that is multiple labels on the same sample, do the dyes overlap? If so, that may cause bleed-through problems between the filters and dyes. So you want to make sure that you use dyes that are separated well enough that you're not going to have a problem. Again, the spectral viewer and other dye choosing tools will be helpful for that. What are the strengths and weaknesses of organic dyes? Strengths, as I mentioned, a wide variety of dyes to choose from. Use them on just about any fluorescence platform as long as you have the right filter sets and excitation labels. There are many live cell labeling options as well as indicator dyes, for instance, for looking at calcium flux or pH in live cells or solutions. We have a lot of reactive dyes that you can use to label proteins of interest, right? So there's a lot to choose from. Weaknesses are that, as with some other fluorophores, organic dyes will photobleach over time as you shine the light on them, and they start to go away. They photobleach. And this leads to a dim signal. And some dyes will photobleach very quickly under certain circumstances, others not so bad. Now you can mediate this by reducing the light, or by using, for fixed samples, you can use an anti-fade mounting heat, which has components that will slow that photobleaching. But it's almost always something you need to keep in mind as a potential problem. Another issue for live cell stains is that many live cell dyes are toxic to cells over time. So if you leave it on too long, you might start to see blebbing or other toxicity-related issues with your cells. So those are some weaknesses. The next class of dyes are the Phycofluors. These are like R-phycocyanin or R-phycoerythrin or allo-phycocyanin, for instance. These are proteins found in algae and cyanobacteria and some marine organisms, some plants that fluoresce, phycobilproteins. And these proteins can be extracted from these organisms and then conjugated to reagents of choice. These really came to light in the early 80s and caught on very quickly. They're very bright. They have quantum yields up at about 90%. They can be excited at one wavelength and detect at a much higher wavelength, so they have a very big Stokes shift that I talked about earlier. And because they're so bright and so highly excited, they can be used in tandem conjugates. That's where one dye will excite another dye so that you can emit at an even higher wavelength. So like RPE, you excite at 488, emit in the orange, and then you have another dye like Texas Red, for instance, that's right next to it. So instead of fluorescing in the orange, that photon now excites the Texas Red, and you emit off in the red instead of the orange. So those are some characteristics of the Phycofluors. They have very high extinction coefficient. RPE is up around 2 million. That's a very high extinction coefficient. Most dyes, as here you can see Electrofluor 647, most dyes are in the tens of thousands to hundreds of thousands. RPE is around 2 million. APC, which is the same wavelength as Electrofluor 647 in the far red, has an EC value of 700,000 and a 68% quantum yield, which is pretty high. So you can see it's much more efficient sometimes to use these phycobilly proteins. You can see some data from flow cytometry and a microarray down. By the way, your second survey question's coming through. I appreciate you clicking on it. But what are the strengths and weaknesses? So I've mentioned the strengths are that Phycofluors are exceptionally bright. They're a good fit for most flow cytometers. For instance, RPE you can excite at 488 or 546. And most flow cytometers will have a 488 laser line. They have that long Stokes shift, so you can do more multiplexing, which is especially important for flow cytometry. And they're good for those tandem dyes. The weaknesses, though, are one of the biggest weaknesses is photo bleaching. Shine a light on RPE, it goes away almost immediately. So it's not good for microscopy, where you need to dwell on the sample for a little while with the light on to get a good image. Flow cytometry, though, the labeled cell just passes through the laser and it's done. So it's just like that. It takes it does the scan, and so it doesn't dwell on it any. So that's why it's mainly limited to flow cytometry. Conjugation is not as easy. A lot of organic dyes, for instance, you can put on a reactive group that will label proteins. It's just a quick reaction and then purify it. But with Phycofluor, you have to do a protein-to-protein cross-linking, which is a little more involved, although we do sell a kit for that. Okay. The third class of fluorophores I wanted to mention were fluorescent proteins. Well, technically, the Phycofluors are fluorescent proteins in that they are proteins that fluoresce. But usually when someone says fluorescent proteins, they mean GFP or RFP or some similar one that can be sequenced and then have the sequence expressed in live cells associated with a particular protein. So it's a transduction method to cause your cells to fluoresce. Well, these fluorescent proteins were first observed in jellyfish, the Cora victoria, which is from up here in the Pacific Northwest. In 1955, Davenport and Nickel found that they saw bioluminescence and fluorescence, and the fluorescence in this jellyfish was from GFP, or green fluorescent protein. In 1962, Shimamura and colleagues first characterized this protein. They actually harvested thousands of these jellyfish, put them in a bag, and squeezed them through the bag matrix to get what they called squeezates, just a gelatin-like mix of proteins. And then they separated out those proteins and found the fluorescent one, and then characterized it. And that was the first real detailed description of the GFP. And finally, in 1992, Douglas Prasher actually cloned GFP and determined the sequence of it. And from there, it really took off. Because once you had the sequence, then you could use transfection techniques, introduce that coding sequence into live cells associated with a particular protein using molecular biology techniques. And then when that protein was expressed, so too would the green fluorescent protein be expressed. And by this way, you were able to determine if your protein of interest was being expressed or knocked down in your sample. Now, green fluorescent protein, the actual fluorescent part is the small molecule that's in the middle of this barrel structure, this tertiary structure. That barrel structure, to some extent, will protect it from the environment. It doesn't stop it from photo-bleaching, unfortunately. GFP photo-bleaches fairly quickly, as do many other fluorescent proteins that have come about since then. But if you try to use an anti-fade mounting medium on it, for instance, you're not going to have much luck because the anti-fade components aren't able to create that barrel structure. Here's a picture of the jellyfish fluorescing, by the way. Quite beautiful. Now, since that initial characterization, there's been untold numbers of papers where where researchers have been able to take that sequence and express that protein in bacteria, in cells of all sorts of different types, including humans. Here you can see mice and zebrafish. There have been monkeys and rabbits. You can even buy zebrafish that are labeled with dyes and sold commercially with these fluorescent proteins called glowfish. In the bottom here, you can see some mice that have been labeled. Those that are green are expressing a particular protein and along with the GFP. And those that are not fluorescing don't have that protein. So you can study if your knockdown was able to move expression of the protein. Here's an example image of some cells that were labeled. We have some human aortic smooth muscle cells, for instance, here that were labeled with our cell light reagent. One for Golgi and with GFP, and one for mitochondria, RFP, or red fluorescent protein. And a nucleic acid stain there in the blue, PERCs. We sell vectors, but we also sell the cell light reagent, which is a Bachmann construct. It's a baculovirus, which is an insect pathogen that's non-pathogenic. And packaged within that baculovirus capsid is the vector that contains the GFP. And in the baculovirus allows entry of that. It delivers that vector into the cell. The cell is transduced, transiently transfected to reveal the next day fluorescent protein, GFP or RFP, along with a protein that's found in a particular organelle, in this case Golgi, and another in mitochondria. Very useful and very easy. You don't have to do the complicated molecular biology techniques for these labels, at least. Now, since that initial GFP characterization, there have been a lot of other fluorescent proteins that have been identified, for instance, corals and other animals, as well as derivatives of GFP. And like, for instance, now you have yellow YFP or cyan GFP. And researchers like Roger Chin, for instance, have really helped in the last 10 years to broaden the palette of fluorescent proteins that are available, ranging everywhere from blue up to far red. And every year they get better in terms of options for fluorescent proteins that won't photobleach as much or are brighter. What are the strengths and weaknesses of fluorescent proteins? Well, the strengths are they're less perturbing to cellular function. Remember, you're expressing a protein. It's not a toxic organic dye, so it's not going to kill the cells as easily or disrupt their normal metabolism. As I mentioned, there's a wide range now to choose from, and you can use these on pretty much any fluorescent platform that you could use for whole cell imaging. The weaknesses are that most of these are not very photostable. They'll photo-bleach away as you're looking at them, and anti-fade components really don't help much with them. Most options involve a molecular biology transfection technique, which, other than our cell light products, really requires some specialized training to do. Transduction efficiency is rarely 100%, often much lower, down like 30% or 20%. It's not unusual to transfect your cells and have to search sometimes for the cells that are expressing the fluorescent protein. Unlike with organic dyes, for instance, which are usually much more ubiquitous in their labeling. Now, you can only label live cells. You cannot fix cells or tissues that have, or you cannot label cells that are already fixed by any means. Although you can formaldehyde fix most of the fluorescent proteins to be retained. However, some of them are a bit fussy, and you can lose the fluorescence under some conditions. For instance, if you fill your cover slip with nail polish, the solvent will kill GFP. And they're not useful for making conjugates like you would with organic dyes or phycoerythrin. You can't just take GFP and conjugate it to an antibody, for instance, with fluorescence. Okay. The final class of fluorophores I wanted to talk about are quantum dots. These are much newer. Only the last 10, 15 years or so have quantum dots really become useful, especially for biological testing. Our quantum dots are called Q dots. These are nanoparticles of a metal alloy that fluoresce upon shining light upon them. They have a very organized matrix of their alloy atoms. And you can coat them to express in a biological assay and give functionalizations. Here's an image with 3 Q dots on it, for instance. 525 on tubulin in green, 585 in yellow for Golgi bodies, and a 655, which is red, for a nuclear labeling. Q dots have been used now for a wide range of assays, cell labeling, flow cytometry. We have a couple Western blot kits now, as well as in vivo imaging. For in vivo, you can inject Q dots, like our Q-Tracker non-targeted, into the vasculature to look at vascularization of tumors, for instance. And one protocol that's becoming much more popular now is our Q-Tracker cell labeling reagents. These are Q dots that are targeted to live cells and are taken up by live cells through endosomal processes, sequestered in the endosomes. So you can track your cells long term. And so you can use this to label stem cells, for instance, and then inject those labeled stem cells into an animal and watch where the stem cells go, and they'll divide and contain the Q dots in them, and you can image them in vivo, including in the far red, or even section them and image them as sections. What are Q dots exactly? I mentioned how they were nanoparticles. You can see here in relation to size with the RPE, it's about the same size. The Q dot 605, for instance, 18 nanometers in hydrodynamic diameter when fully functionalized. The cores of these Q dots are only a few nanometers, 6 to 10 nanometers in diameter. They're electron dense, which means that you can use them for transmission electron microscopy too. And the cores are made of cadmium selenide, for instance. Sometimes tellurium is added in there, or telluride. And then on the surface is a zinc sulfide coating, and this helps protect the fluorescent. And then you can coat that with PEG to reduce stickiness, and a coating to make it more hydrophilic, so you can put it in an aqueous environment. And then on top of all that, you can add your functional proteins, like antibodies, for instance. Now, Q dots are extremely bright, extremely photostable. They blink. An interesting feature about Q dots is they blink. Now, all dyes blink. It's just they blink so fast you can't see it by a naked eye. But the Q dots blink much slower, and you can see the blinking. Take a solution of Q dots and put it under a cover slip. It looks like a starry sky, which is a bunch of Q dots blinking at different rates all over the place. It's not usually an issue because the exposure time that you typically use is long enough that it encompasses the blink rate of the. But there are instances where the blink rate may be slow enough where it can cause a bit of a problem. And there's not much ways to control the blinking. The blinking is actually a good thing if you're doing super resolution imaging, like a single molecule. It fits with the algorithm used in those techniques. I mentioned how Q dots are extremely bright. Their extinction coefficients. Organic dyes usually have an extinction coefficient, which is a measure of the absorbance ability. Right? The more you absorb, the greater the emission that you get out of it. Right? Organic dyes are in the tens to hundreds of thousands. I mentioned how RPE is up at 2 million, which seemed high then, but now compare it to Q dots, which can be up to like 16, even 18 million for their EC values. So they're extremely high absorbing reagents. And they don't have the typical curve that organic dyes have. Remember, I mentioned how organic dyes like Alexa 488 here on the right have bell curves for their excitation and emission wavelength. Well, Q dots are different. The lower you excite them, the more efficiently they absorb. And so you want to use your lowest available excitation filter or laser line to excite them optimally. This is a great thing because it makes for a huge Stokes shift. Remember, that's the distance between the peak excitation and emission. You can have a Stokes shift of even hundreds of nanometers, whereas most organic dyes are only tens of nanometers, times less even. So that means that you can multiplex them much better. I've had for microscopy as many as 6 different Q dots on a single sample. Also, it means that you can excite them all at one low wavelength. So you have one excitation for all 6 Q dots even. There are optimized filter sets for use in your microscopes that will excite your Q dots down low at 350 or 405 nanometers. But you can use standard filter sets too and get an image that is usually just as bright, if not more bright, than an organic dye in the same wavelength. I mentioned how you can multiplex. Again, here on the left is the excitation range for these Q dots. You can see a number of emission curves for the Q dots that we have there. And then on the right is a picture of a vial that contains all of our Q dots in it, different layers, and they're all excited with a blue. This was just a keychain lamp flashlight, so you can excite them all at one wavelength. Quite useful there. You can also combine Q dots with organic dyes. For instance, on the left here we have Q dot 655 with Fluor 488 in the green and DAPI, nucleic acid stain, in the blue. But just as with multiplexing with organic dyes, you also have to be careful about the overlap of the spectra, especially the emission curves. So for instance, on the right we have Q dot 625 and Fluor 594, and you can see the curves overlap. So you're likely to see them both in an Fluor 594 filter set. So you want to avoid that. Here's an example image at low mag, probably just 10x objective, pairing Q dot 625 on the left with a Tritzy labeled sample on the right. Tritzy is a very common orange fluorescent, orange-red fluorescent dye. And so both of these dyes have very similar emissions. The one on the left, you only need a 5 millisecond exposure. It's very low at that objective. Usually it's more like a couple hundred milliseconds. And the Tritzy here, it's pretty bright for Tritzy at 40 milliseconds, but it doesn't come anywhere near the Q dot. In the next one, you can see Q dot 525, which is our dimmest of the Q dots, at a 50 millisecond optimal exposure time. I don't remember what the label is here. But on the right is Fitzy, which is a fluorescein derivative, very common dye that's used traditionally. It took 250 milliseconds to get an optimal exposure with that one. So just some examples there showing how bright Q dots are in comparison. They're also much more photostable. I don't have curves to show you, but here are some example images. Q dot 655 on the bottom, illuminated at high mag for a full minute. You don't really see any change in its fluorescence intensity. While at the top you have Fluor 594, which is one of the more stable, photostable red dyes. But after a minute, it's pretty much gone. Many dyes don't last beyond a few seconds. So anyways, the Q dots are much more stable, and especially if you mount them in our Q-mount mounting medium, which is optimized for Q dots. We've put one of those slides with Q dots and Q-mount on a confocal microscope and let it continuously scan the sample for, I think it was half an hour to an hour. Saw no change at all in the dye intensity. You could never do that with an organic dye. Now, of course, Q dots and organic dyes like Fluor dyes do have different properties. There's no one mounting medium that is optimal for all of them on the same sample. So Q-mount, for instance, will preserve a Q dot signal, but it really doesn't do much in terms of an anti-fade for organic dyes. Hopefully in the future there will be some mountant that will work with all of those. So far, not so lucky. So what are the Q dot strengths and weaknesses? The strengths are, as I mentioned, they're extraordinarily bright. very, very photostable. They have a uniformly low excitation, which is good for multiplexing and giving you that long Stokes shift. They're compatible with TEM imaging, and they're useful for fixed cell labeling. Now you can do some live cell labeling on the surface, or you can do Q trackers to track cells live. Unfortunately, they're not really good for most live cell intracellular labeling other than the Q tracker. So that's one weakness. One person asked about FRET. QDOT has been used as a FRET donor because they're so extremely bright. There are a few papers out there, but we haven't qualified that here internally. Another weakness of QDOTs is that they will quench over time in most mounting media. They're not really archivable unless you use our Q-mount media, which has been optimized for it. In many mounts, the QDOTs will start to lose fluorescence over the course of days, weeks. After a week or two, may even fluoresce very little, only maybe 10% of their initial. In Q-mount, though, it preserves it long term, months if not years, with no change at all. QDOTs, because there's much more involved in the making of them, can be more expensive. So if you need a dye or a fluorophore that has those strengths that I mentioned above, that cost is outweighed by the strength. They are larger size, but it's not usually an issue for most antibody labeling, for instance. They're a little slower to penetrate because of that, so you do need to permeabilize well with using. If you're doing a surface label, the large size has no effect. And then the blinking that I mentioned earlier can be a little bit distracting. Again, it doesn't usually affect your assay then. Okay, so those are the four different classes of fluorophores that I wanted to discuss. Here you can see their relative sizes. Phycofluor and other organic fluorophores are down around 1 nanometer, just a few hundred to a couple thousand Daltons in size. GFP and PE are proteins, so they're larger. And then QDOTs, the more red-shifted they are, the larger they are, but fully functionalized will be 15 to 21 nanometers hydrodynamic diameter. You can see that size in relation to other sorts of cells and colloidal gold. Now this next slide is really the meat of the whole presentation. This is the most important one because it compares some of these characteristics between each of these four classes of organic fluorophores. So for instance, so I'll dwell here for a little bit so you can see it. Again, this presentation will be available later. But the organic dyes, like I mentioned, like Lexafluor or mitotracker green, which labels mitochondria and live cells, fluorescene, they vary a lot in terms of their characteristics. Some are fixable, some are not. Some can be conjugated to protein, some not. Some of them are intended for live cell imaging and will cross the cell membranes. Others are too charged or too large to do so. Photostability varies widely. Many are not photostable. That would only get 1+. A few, like the Lexafluor dyes, are more like 4 pluses. So that varies as well. And pretty much any imaging platform that uses fluorescence will have some dyes that are matched with it, especially in the green. More and more these days you'll see instruments that will detect in the far red, giving you more multiplexability options and vivo imaging options. Fluorescent proteins like GFP, RFP, M-cherry, M-tomato. A lot of the newer ones by Roger Chen have some are named after fruit or vegetables because of their color. You can detect fluorescent proteins with antibodies as well if you do lose their fluorescence. These are intended for live cell imaging and transfection, right? Although they are also fixable with formaldehyde. They're not very photostable, though. You will lose them pretty quickly. And they're intended for whole cell imaging, microscopy and flow cytometry. Phycofluors like RPE and APC and tandem dyes, not for live cell imaging unless you're doing a surface label. They require fixation and permeabilization if you're doing an intracellular label. live cells can be labeled on the surface and fixed afterwards. Again, these are not photostable at all. And the key application, therefore, is flow cytometry. And then finally, Q-dots, like our Q-tracker and Q-dot products. We're coming out with a new line called Q-dot Vivid, which are even brighter than their predecessors. Some of them you can label live in terms of surface labels, and then we have our Q-tracker reagents for live cells, but most of the others are for fixed cell labeling. Some of these products that you can label live are also fixable, like the Q-tracker product. Photostability, 5 pluses, nothing even comes near to the photostability of Q-dots, and they can be used in pretty much any fluorescent application these days. Okay, let's talk for a couple minutes about choosing dyes. There's a number of online resources you can use to help you choose dyes and match them with your filters or to choose new filters for them. One of those is our SpectraViewer. You can see the link here at lifetech.com/SpectraViewer. It's a Java window that will open, and you can compare side by side the excitation and emission curves of up to 5 different dyes and Q-dots at a time, mostly for our products on this. And you can even enter in the specs for your filters, excitation and emission wavelengths, your laser line if you want, and it will overlap it, and you can compare and see, well, does the dye overlap in those filters? Is this filter appropriate for it? You can also look for the overlap in these dyes to determine how good they'll be for fret, for instance, or whether or not there'll be bleed-through between filters with these dyes. Now remember, when I say filters, I'm talking about a little filter cube, usually that goes in your microscope. It has 3 pieces of glass in it. One is the excitation filter, one is the dichroic mirror, like a two-way mirror, and then the third is a emission filter. And using these combinations, you're able to select only a particular dye so that you can get rid of all the extraneous background from other dyes or your light source and get a nice crisp image with good signal background. And you have one filter for each dye, optimally. Take 3 pictures, and then you know, if you have 3 dyes, you take 3 pictures, one in each filter, and then you can overlay those on using your imaging software. Although there are filter sets that you can see more than one dye in at a time, multi-band pass filters. Here, another good resource is our textbook, our handbook, Molecular Probes. This is really a textbook on all things fluorescence. It's a wonderful resource. So if you have any questions about products and don't want to call us here at Tech Support, this is one site you can go to find our online handbook. Or you can order a print version. It's fairly low cost, and you can have it there in your lab to work with. It's an amazing resource. We use it all the time here at Molecular Probes Tech Support. There are a number of resources outside of our company that you can use too. Here's one from the University of Arizona, for instance, where you can find the excitation emission curves of a wide number of dyes, including ones that we don't sell, as well as many other fluorescent proteins, for instance, and some other specs from those dyes. Another good resource often can be found at the filter companies, the companies that make the filter sets. Chroma, Omega filters, Simrock, those are the 3 main companies. Here's a tool at the Omega filters site called the Curvomatic. I love that name. And this is where you can call up a large number of different dyes and see their excitation emission curves. It can suggest filter sets for you, or you can overlay their filter set specs on it and see how they compare with each other. It's quite useful. But these are just a few of the online resources that can be. Another good one is MicroscopyU, www.microscopyu, U stands for university.com. And that site has a lot of good information on microscopy in general and different techniques, as well as the photophysical stuff like this. Okay, well that brings us to the end of our webinar. Here's the contact information for our technical support, where you can reach me and Ed, as well as our colleagues, on questions about fluorescence and our reagent. Feel free to call or write anytime. We also have a chat function. And for those of you who are looking at the recorded version of this, here's some information if you have some follow-up questions. I hope we were able to answer all of your chat questions today. If not, we'll follow up later by email. Thank you very much for attending today. I hope it was useful for you. Be sure to look for further webinars coming down the line. I think we'll have one next on antibody conjugation kits, and then later one for apoptosis assays. All right, thank you very much. Bye-bye.